Part B, structured question on Chapter 2, which is Gene Technology. Discuss the formation of cDNA. So cDNA is actually stands for complementary DNA. So why do we call it as complementary DNA? Because cDNA molecule is produced from mRNA, and the mRNA will function as the template to synthesize cDNA. The purpose of synthesizing cDNA is to produce DNA molecule that has continuous coding region, and this cDNA later you can insert into a plasmid to produce recombinant plasmid. So because cDNA molecule is double-stranded, it can be inserted into plasmid that is also a double-stranded DNA molecule. You cannot insert mRNA into the plasmid, although mRNA has continuous codon region to produce protein, but because mRNA is single strand, uh, it cannot be inserted into the plasmid. So, in order to produce cDNA, cDNA is synthesized, as you know, using mRNA as template. So you will need to identify first of all the protein that you are interested in. So in this case, for example, the protein that you are interested is insulin. So insulin is the hormone produced from pancreas, okay, from pancreas. So in order to get the mRNA, you have to isolate the mRNA from pancreatic cell. You cannot isolate mRNA from another organ, such as, for example, the liver, because the liver does not produce insulin. So no mRNA are present in the cells of the liver. mRNA is only present in the cells of pancreas because the mRNA will need to produce the enzyme insulin in the pancreatic cell. So therefore, by isolating mRNA from the cells that produces the protein, you add the mRNA into a test tube, and then in the same test tube you add the enzyme reverse transcriptase. So for the first answer, which is for the first mark, add reverse transcriptase into test tube containing mRNA isolated from cell. Okay, so once you have added the enzyme reverse transcriptase into test tube containing mRNA, now the function of the reverse transcriptase is to use mRNA as template. So this is for the second mark. So mRNA as the template to synthesize the first cDNA strand. So the blue strand here is to indicate that it is mRNA, and then the green strand here is to indicate that is the complementary cDNA, the first complementary cDNA that is synthesized using mRNA as template. So now there is another enzyme that is used to degrade the mRNA strand, because you don't want the mRNA strand in your DNA molecule. So this enzyme will degrade the mRNA. Next, the next enzyme is DNA polymerase. The function of DNA polymerase is to synthesize the second strand of cDNA using the first strand. So now you have DNA molecule that has a continuous coding region that you can later use to produce recombinant plasmid that you later insert into bacterial cell. State two differences between genomic library and cDNA library or complementary DNA library. For questions that ask for differences or comparison, make sure that every time you answer this type of question, you put it in a table form. to compare the two items or the two topics. So for this question, it is worth 2 marks. So it means that you have to give 2 differences. As for the genomic library, so it consists of collection of all DNA sequences of a species, or you could also write it consists of the entire genome of a species. And as for cDNA library, it contains only the coding region of a genome. So because you know that cDNA, it is synthesized using mRNA as templates. Okay. And for the genomic library, for the second difference is that because genomic library contains all the DNA sequence, so it will take you a long time. Okay, it takes time for you to screen or to find the information for certain genes. Okay, because the genomic library contains all the DNA sequence for that species. So screening process takes a long time. And as for the cDNA library, the screening process is simpler and much faster. Okay, because cDNA library contains only the coding sequence, okay, of a genome. It only contains the sequence for genes, okay, that codes for the synthesis of certain proteins. So that is for this question, that worth 2 marks. So make sure that every time you write the comparison, make sure that the comparison they tally to one another. Okay. DNA fingerprinting is a technique to distinguish different individuals using their DNA samples and commonly used in solving crimes. Figure 2 shows the result of DNA fingerprinting of samples from crime scene and 3 suspects. So this is the DNA obtained from the crime scene, and these are the DNA for the 3 suspects. Okay, so their DNA was analyzed and compared with the DNA that was obtained from the crime scene. So name one example of biological materials that can be used as sample in this technique. So make sure these biological materials that you obtain from the crime scene and also for the suspects contains nucleus, as the nucleus contains DNA or the genetic materials. So common biological materials that will be available commonly in the crime scene, such as, for example, blood, okay, blood stain. So the blood contains white blood cells that has nucleus that contains the DNA, and you could also get DNA from semen, alright, as the semen contains sperm cells that has the nucleus. Obviously, you could also extract out DNA from body cells, okay, any body cells, and then also hair, okay, so hair and also saliva. So those are examples of biological materials that contains DNA. Okay, so make sure in order to get the DNA, they are from cells that has nucleus. Based on the DNA fingerprinting pattern in figure 2, which suspect present in the crime scene? So in order for you to identify which is the suspect that was present during the crime scene, so you have to compare the DNA pattern, okay, the DNA bands, the patterns of the DNA bands. So for the 3 suspects, as you can see, suspect 3 has the same DNA bands, the patterns of DNA bands as the crime scene. So as you can see, so the DNA pattern is the same. Okay, so this will conclude that suspect 3 is the one that was present during the crime. Okay, and then for the next question, explain how you decide which suspect that you have identified for question number 2. So basically, for this question, it is worth 2 marks. So as I said, in order for you to identify the suspect, okay, so you have to compare the DNA patterns. Okay, compare the DNA patterns between DNA from the crime scene and also the DNA of the three suspects. That is for the first point. Okay, so for the second point, make sure that when after you have compared, in order for you to identify the suspect, make sure that the DNA pattern of the suspect matched, okay, with the DNA pattern that you have obtained from the crime scene. So if they matched, so it means that the suspect is the culprit here. Okay, so that is for the second point.